I'm Daniel Levine, and this is the Bio Report. A chronic shortage of high-quality targets remains one of the biggest constraints in drug discovery, even as therapeutic tools become more powerful and diverse. Regeneron is tackling that problem with its Regeneron Genetics Center, which has built a genetics-driven discovery engine that integrates human genetics with rich clinical data, large-scale proteomics, and AI-driven analytics. Aris Baras, head of the Regeneron Genetics Center, discusses how proteomics is reshaping RGC's view of risk prediction, how AI helps his team sift through hundreds of millions of variants, and what it takes to scale this kind of effort and translate it into more successful, transformative therapies for patients. Aris, thanks for joining us.
Thanks for having me. Looking forward to it.
We're going to talk about the Regeneron Genetics Center, its evolution, and how it's shaping drug discovery at Regeneron. You've led the RGC since 2013. How has the original vision of linking genetics to therapeutics evolved now that you've sequenced millions of exomes and built a biobank scale platform?
Well, thank you. My favorite topic. It's a wonderful team here, first off. So the team gets all the credit. It's a couple hundred people. We're some of the best in the business who've been doing this for over a decade, as you mentioned. And it's an interesting question, how have things evolved? Because in one sense, the core concept, the whole core mission that we started this around is still central today. On the other hand, so much has changed around that and how we do that. So let me elaborate a little more. When I mentioned the core mission here, we set out to address a big problem and a simple one. That was the target bottleneck. We felt at the time that we had all these amazing modalities, certainly an antibody therapeutics company and all these amazing technologies around us. But one thing we were struggling with, and so many people were struggling with, is what are the great targets? And you've seen this. Anytime there's a great target, there's a race of dozens of companies and therapeutics going after that same target, right? And a stat that I like to throw out there, and this is going way back, about 10 plus years ago, but we actually added up all the genes and targets for every medicine that had been approved or for any experimental medicine that had been tried. And it was only about 10% of the genome, right? Meaning there was so much space, targetable space that we hadn't explored. There was so much we could learn about targets. So that was the simple concept. We were inspired by just a few amazing genetics stories, some human experiments that led the way to amazing therapeutics. And those were of a couple varieties, but one I really want to talk about are protective genetic factors. Many are aware of the PCSK9 story, people who had genetic mutations that break the gene basically, and they have 90% lower rates of heart attacks. There was another amazing story around HIV, people that were missing a functional copy of the receptor on your immune cells that HIV uses to infect your immune cells and wipe them out. And these people couldn't get infected by HIV. And a couple companies made great medicines going after that CCR5 receptor. Danny, that's what we were looking for. We were looking for more of these types of examples. So I can tell you more about what we have found there. The evolution piece is we learned that you really have to scale this thing. Maybe naively, but we thought, hey, let's sequence 100,000 people and we can do some damage. And fortunately, we had a few important discoveries to show for it. But what we learned was you really had to hit this at huge scale. You're looking for needles in haystacks, things that are, 1 in 1,000, right? Or even more rare. And then to be really sure that you've found something and it's real, you want to see that hundreds of times or thousands of times to be convinced statistically that you're going to take this thing forward and invest hundreds of millions of dollars behind drug development campaigns. So that's how you get to sequencing millions of people, So that's where we are today. We've sequenced millions of people, as you mentioned. We have pretty confident we have a path to get to 10 million people sequenced. And it'd be fun to talk today at some point about a new ambition we have, which is how do we get to 50 million and 100 million people, and why would we do something like that?
You are doing exomes as opposed to whole genome sequencing. As you're looking for those rare genes that might lead you down to a novel therapy, why focus on exomes as opposed to the whole genome?
Great question. And that too has evolved, to be honest. We started with exomes for two big reasons. By definition, the exome is what all your genes are, what encodes all your genes. So for those who aren't familiar, your genome is this big bad, 3 billion base pair thing. And then it's only about 1 or 2% of that where your actual genes exist. So we wanted to focus where the money is, where the large effects are, if you will. And when we got started, there was a big, big cost problem. So this was an expensive scientific experiment or expensive endeavor. And the difference between an exome and a genome was huge. It was 10 times the cost. So you have a tough decision to make. We just talked about how important it was to find those 1 in 1,000 and study them 1,000 times over. So the million person scale problem, that becomes 10 times more expensive. Or if you're doing genomes, you do 10 times less, which is really problematic. So at the time, we focused on exomes, but part of the evolution is that technology has come such a long way, and the cost of sequencing over this last decade has come down tremendously. And in fact, we do a ton of genomes now. So that's not as big of a problem, and many of our projects moving forward are very large-scale genome projects.
You've been at this for more than a decade. What's surprised you? What's been harder than expected? What have been some maybe unexpected benefits that you've seen from this work?
The hardest thing has been scaling, honestly. If you think about how this all comes together, we've got to find these great partners out there, these health systems, medical centers, great passionate scientists and patient advocacy groups, and put the whole thing together to get hundreds of thousands and millions of people to consent and volunteer and join these studies. It was quite successful to go from nothing to hundreds of thousands or even the first few millions that we've been able to sequence and have them be part of these studies. It's been really challenging now to go to that next level. But I'm optimistic. There's a lot of exciting things going on there. There are some groups that are doing some pretty ambitious, amazing, heroic things. You can look at what the UK continues to do. They've got a wonderful project, Our Future Health, working across the entire nation and health system there, and well on their way to getting 5 million people into that study. There's the US effort as well. We've been working with some groups like Truveta and their 10 million person project. But there's something new that we're also trying. We're trying our hand out, again, because this is harder than we thought it'd be. So the old school model has been what I just described. Something new we're trying is, can we uncouple this process, right? This kind of one-stop shopping where you go to these health systems or you go to this major research group and you do everything. You do the consenting, the biobanking, the health data collection. And some groups have been trying to say, hey, let's aggregate all the health data, right? Let's go get hundreds of millions of health records, de-identify them, compliant, privacy preserving, and let's aggregate that in really high quality. There's other groups out there that are working on, let's get really large scale consenting and biobanking and sample flow. So can we uncouple these things, right? So now you can move at kind of tens of millions, hundreds of millions people scale. And then when you get consent and permission from people to join studies or as part of clinical care, you can then link and merge these things together. So you will have seen us over the last few years done some major partnerships, pretty significant investments, $100 million plus type of investments with Truveta, more recently with Trinetics. And that's what that's all about. You also asked what things came a little easier. What came easier actually was the pace of discovery. So many of us here remember very vividly when our CEO told us, hey, I'm all in on these projects. Let's do this in terms of human genetics, and let's sequence hundreds of thousands of people and get to millions of people. I remember him saying, let's just find one more PCSK9. If we find one more PCSK9, this is all worth it, right? So it might surprise you if I told you that we have found over 50 of those PCSK9 type of protective genetic factors. We call these superhumans, right? These people that have these rare mutations that protect them from disease. PCSK9 happened to be with cholesterol and heart attacks. What we have found since has been dozens of examples, not just in heart disease, but in obesity, in blinding eye diseases, in neurologic disorders. We've even found them in cancers, people who are born with mutations, variants that protect them from getting cancers. It's pretty remarkable. I guess the last thing you asked about was side benefits, things that we hadn't expected. The thing we hadn't expected that's been a huge side benefit is how powerful these data are for predicting health outcomes. Has nothing to do with drug discovery and development in a sense, finding that target, that needle in a haystack. When we started to look at clinical trials and sequencing people in our trials or sequencing people in these large health system studies, we realized that it's not a single gene, but looking across their whole genome, you can really get an accurate prediction of what types of future health risks people may have. Now, that has enormous value in clinical trials. If you're running a clinical trial and the endpoint is heart attacks or neurodegenerative disease, it's really important to be able to stratify patients and run efficient trials. But outside of drug development, think about the value to society and to patients and to providers. If you can take people who are, seemingly well and don't have disease, and we could just understand what cancer they might have in the future or what heart disease they may have in the future. We all know how powerful early detection and prevention is.
You talked about the success you've had with identifying protective genes. I imagine one route to getting to that is through the study of monogenic diseases. And I'm wondering how you think about your work in terms of monogenic versus highly polygenic, multifactorial diseases when deciding what to focus on.
You're right. I mean, it's really changing in front of our eyes how we think about disease and how we classify disease. So monogenic, as you described, has been, I think, the bread and butter of human genetics since its beginnings, right? And Regeneron, like many other companies, really pursued that with a lot of vigor. So some of our earliest successes at Regeneron are going after rare genetically defined diseases. So that's really at our roots, and we've done that, over and over again. In fact, just last week we had a wonderful example of that again. Another monogenic disease, one that's congenital hearing loss, and in this instance caused by a genetic deficiency of the otoferlin gene. So luckily it's not that prevalent. So there's only 50 or 100 kids a year who are born with this. But to your point, when you understand what's causing it, you can imagine doing things like, hey, let's try to correct this or restore its function. And so we developed with our collaborators a Decibel that about 2 or 3 years ago, as part of an acquisition, became part of and integrated into Regeneron, a gene therapy that can actually deliver that missing gene right to the hair cells that need it in the cochlea. And as a result, most of these kids are restoring their ability to hear, and many of them in the normal hearing range. So that's a medicine, a gene therapy that was just granted accelerated approval last week. So this is a huge part of what we do in terms of continuing to pursue monogenic diseases. But your other point is that, the contribution of genetics to disease is far greater than what we thought in a lot of these single gene disorders. And we're realizing that a polygenic contribution, to use your words, underlies so many different diseases, right? And we can, just like you can find that cause of hearing loss and it's otoferlin, or you can find the cause of some other disease and it's a single gene, we're finding that you can find equivalent diseases of a very significant, huge penetrance, huge disease risk, and it's not driven by a single gene, right? It's driven by a collection of many, many, many other smaller risk factors. But added up, they can be just as profound and just as significant disease risk as that monogenic disease. So yes, we're seeing that everywhere, whether it's heart disease, obesity, fatty liver disease, neurology. That concept is everywhere and is really redefining how we understand disease.
You've expanded beyond genomics into proteomics and other omics layers. How are you integrating these data sets today, and what additional insights do they unlock?
This is one of the biggest surprises we had here. Proteomics is something that when we talk about it, so genetics, we had this amazing conviction, right? We talk about Regeneron being a genetics company at its root, at its core. And so this was an obvious thing that we wanted to do. And then we looked at some of these other data types, and I forget exactly how it happened, but we got presented this opportunity to get involved in proteomics. And the first big project was called a pilot project, right, with UK Biobank. And it was 50,000 individuals where we did a whole proteome, if you will, right? It was thousands of different proteins that were measured. And we didn't really know what to think about it, honestly. We thought we'd seen some really compelling and interesting data elsewhere. We thought the general concepts like, wow, being able to look at so many different proteins and biomarkers across so many disease states could be really interesting, right? But we far underestimated the utility and the power of proteomics. And we got our hands on this first 50,000 person study and data set. And so kudos to, the many organizations and scientists who championed this. Kudos to Rory Collins and the UK Biobank for being so persistent with this. And Danny, the power that the proteomics data held was, it really amazed us. So a couple types of examples. We just talked about how predictive human genetics is, right? We get this genome that we inherit, and you can predict so many things, not to rehash them. Your cancer risk, all of the disease risk. And then we asked, what about the proteome? What could the proteome tell us above and beyond that? And in most instances, it was actually telling us more. It was able to predict more about your disease in the next year or in the next 5 years than the genome was. And it was dynamic. Your genome was something that is something that you inherit, and your risk is more or less the same from your inherited genome, but your proteome is dynamic, right? Every year of your life, every time there's a disease state you might encounter, the proteins circulating in your body are changing dramatically. And their ability to tell you that, to give you signal, is remarkable. So, we were immediately enamored with proteomics, and we scaled up significantly. So now, we, with a bunch of other companies and back with UK Biobank, are finishing the project, are doing the entire UK Biobank. We at Regeneron have also been rolling out proteomics at population scale, health system wide, with many of our health system partners. So we've now gone from that pilot to the hundreds of thousands of person scale and moving to the millions of person scale in proteomics.
If you think about RGC as a discovery engine, what are the primary outputs to the rest of Regeneron today? Are they new targets, biomarkers, patient stratification strategies, or something else?
All the above. Targets was the founding idea, right? There's a bottleneck in targets. If we can get some of the best targets in the industry It'll be a great competitive advantage for Regeneron, be great for patients, greater probability of success. And all that has borne out, right? We've been able to put, I think I mentioned beforehand, 50 new targets into the Regeneron pipeline. Not discoveries, but real bona fide programs with molecules behind them and investment behind them to move them hopefully to the approval line and to patients. So targets has always been a big thing. Another way we work is we take existing drug development programs, and there are a series of really important questions that these types of data can help guide a development program. So we might look at patient stratification, as you mentioned. We looked at a great example, which was cardiovascular outcome studies. I mean, it's the gold standard, right? To show that a drug is going to do something, a new medicine is really going to have benefit on cardiovascular outcomes and mortality, and to get patients and society to understand what the benefit is and to get comfortable to really use it. But boy, are they expensive, right? It takes a long time. You need a lot of people. I think we had 20,000 patients in our cardiovascular outcome study for our PCSK9 inhibitor. And the unfortunate thing is, out of those 20,000 good for them, right? That there's not a very high event rate, and we're not having a lot of heart attacks for those individuals in the placebo arm, for example. It's about 10, 11, 12%, right? So most of the study is not having any events, and it's somewhat wasted. So to see the types of 30% or whatever benefits we're seeing, you have to have massive studies. So we put a paper out there that others have done similar things with these large cardiovascular outcome studies. And we've shown that you can use genetics and other biomarkers to dramatically improve this. You can identify subsets of those 20,000, for example, who were already selected to be at-risk patients who've already had cardiovascular disease. But now you can identify people at really high risk. We call them high-risk, potential high-benefit patients. And you can find people who have a 30% chance of having a heart attack in the next 3 years, right? Not 10%. So we do that for all of our programs, right? Whether it's COPD and you're looking at exacerbations as the endpoint there, or you're looking at fatty liver disease and you're looking at advancement of fibrosis and cirrhosis, and on, right? Whatever the endpoint is, oftentimes these endpoints are a bit more rare, and you would love to stratify and enrich for those. Another really valuable thing that we do using these data, we try to find additional uses where there could be benefit. So look at some of the most successful medicines that the industry has been able to develop and bring to patients. A lot of times these medicines can be really helpful to patients with many different diseases. Take one of our own medicines that is an immunologic agent and goes after this kind of Th2 type of immune response, blocks the IL-4 receptor. And the genetics have been very clear, and the clinical trials have been very clear. It all lines up. There's multiple clinical settings where there can be benefit, whether that's asthma, whether it's eczema, or other indications. And so a lot of times we find ourselves with a really hot start, a great target, a great molecule and potential medicine, and it's got its lead indication or disease that we're studying. And we can go back to genetics now and ask, what other diseases could this be useful, right? And so how do we do that? You start with, here's the target, and we've got a drug that blocks that target. And so we can go into our human genetics databases and find individuals who have knockout copies of that gene, right? Or loss of function, as we call it in genetics. And that's basically a surrogate or a mimic. And now we can scan the whole database, not just in that initial disease, and confirm, yep, people with loss of function are protected or have lower risk or do better when they have this disease that we already knew about. But now we can ask that across thousands of other diseases. And oftentimes we find another or 2 or 3 more areas where there's really strong evidence. And then we can go launch trials in that space as well. So we call that kind of indication selection, indication expansion. So lots of valuable ways, as you mentioned, from targets to stratification to new indications.
What does it take for you to decide a genetic association is strong enough to advance a target into discovery pipeline versus keeping it as a biomarker or a risk signal?
We're getting a lot better at that. So I think that's something that the whole field has gotten better in this last decade with this mega scale sequence data. For us, it's really been the same. We started, as I mentioned, over 10 years ago with the simple concept that we wanted really large effect human genetics findings. So we wanted to see things like the otoferlin example, where individuals who have that genetic deficiency, it's like 100% penetrant, right? You know, they won't be able to hear. It's really powerful. And if you're able to restore that function, there's a really good chance you'll be able to really improve or correct, that disease. On the flip side, if you've got a protective variant, we really want to see something that, hey, if it's going to have an effect on weight loss or obesity, like our GPR75 discovery or the GIP receptor discovery, these things are going to lead to, 60% or more in terms of lowering the risk of obesity, or 20 pounds or less average weight than the average person type of thing. So we've really shied away from, and the other thing is really having a clear sense of the direction of effect, right? We really need to be clear from the genetics that, hey, this is a loss of function. The gene has been broken or inactivated. So it's rather obvious that if you want to mimic that effect, you have to make a drug that's going to block, right, the target. Or if the variant's going to rev up the gene and its gene product, right, or an activating or a gain of function, as we call it. So then we know we've got to make a medicine that's going to be an agonist, right? So those are two pretty critical things. And you can get lost in genetics a bit, right? I mean, genetics is a huge field. There's a huge amount of signal there. And up until the days of very large scale sequencing, and especially focusing on the genes themselves, most of the genetic hits have been these, what we call GWAS hits, or a lot of these associations that the actual genetics are outside of the genes. They're way outside of the genes. And the effect sizes are really small. They're 1% or 10% at best, right? Compared to the, 60% and 100% that I was telling you before. And that's a problem, right? Because if it's a really small effect, then what's your therapeutic going to do? If it's not clear what gene is being impacted, it could be impacting multiple genes, then what target do you go after? So that's why we've been really disciplined around focusing on very large effects and focusing on clear signals in genes where we know what the direction is. The rest of the game is, a more traditional drug development challenge. Just takes really savvy and experienced drug developers and understanding, okay, where does this target play a role? What cell types? Is it inside the cell? Is it outside the cell? So what type of modality do I have to have? An antibody or a genetic medicine or a small molecule? And then what's the path ultimately in terms of clinical development? How do I do trials to get proof of mechanism, proof of concept, and all the way to approval?
You touched on modalities. Regeneron works across modalities, but I'm wondering, now that we can come at genes and genetic targets and various points along the translation of a gene into a protein, how does that shape your thinking about modality selection?
It's really shaped our thinking. You've seen us evolve and reinvent ourselves over the last decade here. For very good reason, Regeneron really developed as a company and kind of rocketed up, focusing on protein therapeutics and actually more specifically on antibodies. Amazing technology developed here way before my time around engineering the mouse genome so that it had a human immune system, right? I mean, an amazing story for a whole podcast in and of itself. And there was immense focus at becoming really an antibody leader there, right? And being able to use billions of years of evolution to be able to immunize mice and make these amazing human antibodies. And we did really well doing that. And here's what happened with human genetics, or this, what I'll call kind of explosion of discovery, right? And target opportunities. So it really worked. And one key fact that I guess we all knew, but it wasn't top of mind, front of mind when we jumped into this. When you look at all those genes, 20,000 genes or so in the genome, there's only about a quarter of them, or a third of them, for being really generous, where the gene product, or the protein they encode, is available on the surface of a cell or is secreted and gets outside of the cell. In short, something an antibody or a protein therapeutic could effectively engage, right? And what that meant was there's 2/3 or more of all these targets that are not accessible or tractable by an antibody therapeutic. And so, we learned this the hard way as we were making all these discoveries, and we could funnel some of them where appropriate to an antibody approach. But so many of them, we were realizing, gosh, we can't get at this amazing target we're really excited about with an antibody. So it really forced us to reinvent ourselves and think about, what is our strategy going to be? Are we going to pass on those? Or are we going to proactively go into that space? And that was a really defining moment for Regeneron, and a lot of tough decisions. So we had to think about, do we get into small molecules, something we knew nothing about? Do we get into genetic medicine modalities? What would be the play? What we decided to do was we invested heavily in genetic medicines. Why? There was a lot of properties there that we liked that were very similar to antibodies and were core principles for us, right? They were precision medicines. So these things you can literally, as you know, design, right, by their sequence. Just like antibodies are really precise. They go after their target and hopefully nothing else. Small molecules, amazing medicines have come from that, but it's a little bit of a messier process, and you don't have that type of precision and specificity. Speed was really important too, right? With antibodies and genetic medicines, you can very quickly go from a target to a lead candidate, right? That's not exactly the same with small molecules. So that really got us into an amazing new world for us. We did some phenomenal partnerships that have produced a lot, for example, with Alnylam. And that was, again, around marrying their technology, world leaders in siRNA technology and capabilities, and marrying that with the output and human genetics target discovery we were generating at Regeneron. And it was a really ambitious partnership. John Maraganori and the wonderful team at Alnylam and George Ankopoulos and our team here really had this vision of a long partnership and rich partnership together, and a big one. Things we're talking about, I think 30 or more programs that can emanate out of this partnership and this marriage of these two approaches. We are, time flies, so I don't know exactly. It must be year 5, 6, or 7 in that partnership. And it's been almost exactly that. I mean, we've had 20 plus programs come out of this marriage of great human genetics targets with best-in-class siRNA capabilities. And we did a couple other things too. We got into genome editing, right? That was a hot field and has a lot of capabilities that you can't accomplish with some of these other modalities. So we've done some really exciting programs there. And we also have invested substantially in gene therapies, as we talked about with our first program, which was the hearing loss one. And one common thread across all of these for Regeneron was, we talked about why we got into these, but another core principle for Regeneron is that we really like to contribute in terms of technology development with our collaborators and to work on the next generation of technologies and platforms that are addressing today's biggest problems. And in the field of genetic medicines, enormous credit given to the Alnylams of the world, to those who've brought forward the editing platforms, for example. And what Regeneron is able to contribute is not just the human genetics targets, but now how can we help deliver those genetic medicine cargoes to the next frontier? A lot of these things started as delivery to the liver, and because that's where they naturally can go and be taken up. And let's go after a ton of diseases of the liver, or targets that are expressed in the liver. And we've been working now on, okay, let's target those things over to the CNS and the brain. Let's target those things to the muscle, to heart, to kidneys, to lungs. We've had some good preclinical success. So we actually have platforms now on the first couple of ones I mentioned, getting things to the brain and CNS and to muscle. And there's enormous exciting opportunities there. And we're working on the next generation of those targeting platforms and all those other organs and tissue types that I mentioned as well.
You're dealing now with massive data streams, whether it's genetics or proteomics or patient records. How are you using AI and machine learning to help you get to actionable decisions around biology?
In a few pretty specific ways. We've been actually using machine learning, as you mentioned, and broader AI tools for quite some time. If you think about some of the workflows and data science that goes on here, we look at, so when you're doing sequencing and have human genetics and we talk about the end product, we simplified, make it sound pretty easy and say, hey, we discovered this variant, this gene has a huge impact on this disease. What we end up doing is having to interpret and sift through hundreds of millions of variants that come across when you sequence millions of individuals, right? The amount of variation that exists in everyone's genomes, especially when you look at it at such massive scale. And which of that variation matters at all, right? Most genetic variation, fortunately, doesn't actually do anything. It doesn't harm us. It doesn't give us these superhuman benefits. It's just kind of variation that happens. There are error rates. It's part of evolution. Mutations happen, and most of the time doesn't do anything. And there's some clear-cut things. I've talked about knockouts and loss of function. If there's a mutation early in that gene and it creates a stop, all right, it's pretty obvious what it's doing, right? We know that basically turns off the gene entirely. But there's everything in between, right? There are so many variants, and it might change the genetic code by a base. And we don't know what exactly that does. And so we've been able to use AI to help us understand that. And we've been doing that for years. And how do you do that specifically? Well, you need great models. I think we've all learned that. You need great AI models. But you also need, the other part of the equation is great training data. And when you have millions of exomes and genomes like we do, we can learn from all the other genomes we've done. When you have all this amazing proteomic data, right, you can pair that. And yes, you can confirm when that one stop mutation that I mentioned, we say we're pretty sure that it turns off that gene. And then, no surprise, that protein, we can't find it, right, in that person's blood sample, right? It wasn't produced, right? It's nice confirmation. But for all the harder stuff to interpret, that one base pair change, there are examples where that one simple change also turns off the gene and we might have not predicted it, right? And then you can see it in the protein level drops, right? Or it might dramatically shoot up that protein level. We can look at the associations with these variants and all of our clinical data, right? And which variants are associated with disease outcomes, right? So that's the kind of data we have that we can point these AI models toward. And in short, it makes our ability to predict which variants are bad or good or benign much, much better, right? And same thing, we do this with imaging science, for example. We've got access to hundreds of thousands of images. It's an amazingly fruitful area of research and discovery. And we can automate the detection of certain features in images that might correlate with genetics or disease. So AI has been an area that's been a core for us in terms of our basic discovery abilities. We've relied on that. We've needed that. And increasingly so, as now we think about other areas beyond drug discovery and development, as we talked about earlier, the predictive power of these data, that's another place that we're deploying AI. How can we help speed up the interpretation or the reporting back to people of massive amounts of data It trains and learns on healthcare outcomes. How can you, for example, get the next patient's data or upload your data and get, incredibly accurate predictions about, what your future risks of disease might be in your health outcomes.
And has the use of AI changed the culture of discovery within RGC or Regeneron more broadly?
A little bit. You know, it certainly has sped up a lot of things that are operational and administrative. So when you think about some concrete examples like coding, um, it won't be a surprise to you that there's enormous amount of coding that goes on in a place like the Regeneron Genetics Center. We have massive amounts of data. We have an insane amount of compute and analysis that goes on, which means people are creating pipelines and algorithms to run on this data. And that used to take armies of programmers and data scientists to do all of this. And we still have a lot of people that do this, but it's been really amazing. You're familiar with how these platforms have been so good at assisting coding. So we use that and it speeds up the amount of time we spent on coding and implementing our platforms and pipelines tremendously. So we really use it. Oh, and then other examples would be we find these discoveries, right? And then we need to pull together all the information we can about what is known about that gene, right? And we start to fill in all those pieces we talked about, like where is that gene important? What cell or what organ? What is known about it? What models have been studied? And, not an insurmountable problem, but it used to be a lot of people hours and people time to pull that information together. Still requires, smart minds and human supervision. But the ability for these tools now to do that way more efficiently than we used to and pull this all in and synthesize it and summarize it for us is really remarkable. So those are just at least two examples of how that's changed our culture and our speed to go from, data to discoveries and discoveries to actual, target hypotheses and things we can now invest in and put research plans and development plans around them. But it's still very much, this human inventive culture around here and the passion of the scientists and the teams at RGC that cook up, these ideas about let's go after protective superhuman genetic factors, right? Let's be ambitious and, 13 years ago and try to sequence hundreds of thousands, or now the evolved version of that, let's try to get to 50 million or 100 million people for all the reasons that we have. So the culture is really shaped by both of those factors, right? The kind of the human inventive spirit and passion and the core mission behind this all, as well as what a pleasant development with all these tools that can assist us and help us and do a lot of the operational grunt hard work a heck of a lot faster. And boy, do we need it in time because the scale is just getting bigger and more challenging. So it's the right time to have these tools assist us and help us.
And as you think about the company's pipeline, how confident are you that the work RGC is doing is increasing the probability of success? And how do you measure that internally?
It is increasing the probability of success. It's actually something that's pretty easy to look at. You can look at probability of success metrics across the industry. There are great databases that track this kind of thing, and you can look at probabilities to go, from entering the clinic to approval, or if you want, you can look at, some smaller phase from just getting into the clinic to proof of concept and anything you want, right? So the numbers are really good to know what does it look like in the past or what does it look like across the industry. And you can get specific about what types of targets do they have human genetics evidence or did they come from other places? Or even what diseases, like is cancer easier or harder than neurology type of thing? And then we have our own data, right? Almost 40-year company and experience, a pipeline that has over 50 programs in it, I believe at the moment, and a lot of good data. And so we can look at all the different phase transitions. And I'm pretty sure we've talked about this publicly. We actually have higher probabilities of success in all those phases, right? Going from getting into the clinic to getting to proof of concept, to having successful phase threes, to getting approvals. And a large part of that is due to the fact that we select a lot of our programs based on human genetics evidence. So even if you normalize things to other antibody programs across the industry, or even if you compare things to apples to other genetic medicines programs, when you select human genetics targets, and especially of the types that we have, these large effect, clear direction of effect, it's a really high probability of success. Some other groups, Danny, have published on this, groups from GSK in the past, some academic groups. I think the industry number is that 90% of things fail that go into the clinic, right? Or on the other side, only 10% of things that go into the clinic make it to the finish line. So people have shown kind of hard, rigorous data that can be 2 or 3 times greater, depending on what your definition is of human genetics evidence. And I think in our hands, we've seen even better than that, right? It can be 4 or 5 times higher success rates when you're starting from these types of clear, large effect, protective genetic factors. So, I mean, it's really transformative, right? If you think about changing something by 2 or 3-fold, or maybe even 4 or 5-fold in terms of the success rates, when you don't use human genetics, it's a no-brainer, right? I mean, it's really game-changing.
As other companies might think to replicate what Regeneron's doing, how easily is that done, and how dependent is it on your ability to do this at such a scale and integrated level?
Well, definitely people are doing this, right? We are not alone. There are some wonderful companies and leaders and programs out there. Maybe when we started, there weren't as many, but now there are certainly many. And we actually, on the one hand, really appreciate them because, they're not just competitors. Many times we collaborate together to make some of these mega large-scale projects happen. UK Biobank's something that we all benefit from. Now it would be something we could say, sure, maybe Regeneron, we can do that on our own. But back then, this was many years ago, to sequence 500,000 people at that cost on our own might have been prohibitive. So having all those companies come together and share the load. Now we did the project, but at least to share the cost and do it together was really transformational. So we do that a lot with companies for some of these, mega projects. Like I mentioned, Our Future Health and that 5 million person project, that's probably going to require a lot of companies coming together again. On the other hand, we're competitive and we aspire to be the best at this. And it's a big competitive advantage for us. So the things that we're focusing on are absolutely scale, as I mentioned. At the moment, we do believe we have the largest scale in terms of the size of the database and the richness and depth in terms of longitudinal health outcomes tied to it, new dimensions of data that you asked about, proteomics. And, we might be alone in terms of our aspirations now to get to tens of millions, right? We've talked about, we want to get to 50 million people. And I don't know if others are really going to go there. The other place that we want to differentiate and create an advantage is in our analysis and analytical capabilities. And that goes back to being in part the people, and back to this point I mentioned, the team here, give them all the credit, and they're phenomenal. We have over 10 to almost 15 years recruited and attracted some of the greatest talent in human genetics and statistics and data analysis. I mean, we are incredibly fortunate and privileged to have those folks, as well as the technologies and tools from AI and machine learning. I mean, we are, as I mentioned, very early adopters of that, right? And very heavy adopters of that. And maybe the only other thing I would add is our ability to so quickly translate and traverse that bridge from human genetics discovery to therapeutics programs, right? I mentioned we have 50 programs that we have discovered here and now created actual drug development programs at Regeneron. And it's gotten to the point now, because obviously that's scaled, right, with more data and more analysis going here, it's been more than linear. And so we're putting about 10 new programs into the pipeline every year from new discoveries here. And that's a Regeneron thing, right? Goes back to your point about culture. It goes back to the core mission, the fact that genetics is in our roots and that we value and believe in this stuff so much. So our ability, when that discovery, hits the analyst's desk, so to speak, that day, there's others here, I'm seeing it that day. We're getting excited about it or not if it needs, some more work. And that chain happens quickly, right? And then our research teams and our therapeutic modality teams, it's instantaneous. It's like a, chain reaction. And we're moving very quickly to approve significant investments and programs and campaigns. People are primed and ready for this kind of thing, right? To go from genetics discovery to a bona fide, target and therapeutics campaign. So I think those are the ways that we try to stay ahead of the curve and try to have this be still a major competitive advantage for Regeneron R&D and for Regeneron as a business.
Aris Baras, head of the Regeneron Genetics Center. Aris, thanks so much for your time today.
Thank you. Pleasure was all mine.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.